Hello, participants, and thank you for attending today's webinar. We welcome your questions. First of all, please do not post any questions using the chat. Use the Q&A button that we have at the bottom of your screen. You can also vote on questions from time to time. We're going to wind up having some polling in the Q&A that you would like to have answered. As I said, please do not use the chat for the purposes of Q&A during the next couple of hours where you're joining us here. Answers to questions will be published after the webinar, along with the presentation slides and a YouTube link. So we want to make sure we're disseminating this information as widely as possible. Good morning. My name is Mark Johnson. I am a material scientist at Clemson University. I'll be co-moderating this session today. My co-moderator is Marianne Tung. Marianne, you can introduce yourself real quickly.
Hi, everybody. Welcome to the webinar. My name is Marianne Tung. I'm an electrical engineering PhD student at Stanford.
Great. Thank you, Marianne. So with that, I think there's a lot of very important information we want to get to today. We're going to begin by posting a poll on your interests in this event today. So if you can look on your screen, you'll wind up seeing a poll. We'll take a couple of minutes while this poll is going on, just to make sure that everyone's able to wind up logging into this meeting today. This is great response. Thank you all for contributing. It gives us a great understanding of where you're coming from and how this information can potentially be of value to you. So we really appreciate having this information. And following the chat, seems as though we've got a number of people joining us from around the world. So thank you very much, all of you, for coming in. This is terrific to see as we wind up working together and addressing all these issues. I think we have another 30 seconds or so on the poll, and we'll wind up getting going. So thank you very, very much for your inputs. And as I said earlier, for those of you who have recently joined us on this in the last minute or so, thank you for attending. First of all, thank you for joining us. We welcome all of your questions. Please post any questions you have in the Q&A button. A number of questions have already begun coming in. The Q&A button at the bottom of your screen. Please do not post questions in the chat. Answers to all these questions will be published with the webinar along with the presentation slides at the end of the video link. With that, I think we're going to wind up kicking off our event today. I'd like to welcome our first speaker, Hannah El-Samad. Hannah?
Hello, good morning, everybody. It's morning here in California where I am. My name is Hannah El-Samad, and I am a professor in the Department of Biochemistry and Biophysics at the University of California, San Francisco. It is truly my great pleasure to welcome you all and introduce N95DECON, one of the co-organizers of this event. N95DECON is a consortium of more than 100 scientists, engineers, medical doctors from more than 10 universities, as well as other professionals who are working together on a voluntary, non-paid basis to address one of the most pressing issues of this pandemic, which is the shortage of personal protective equipment. Our motivation was specifically the shortage of N95 respiratory masks. We were propelled to action in March 2020 by reports that healthcare workers, both in the US and throughout the world, are experiencing a shortage of this crucial protective equipment and the increasing need to fill the gap in their decontamination and reuse. So we came together as a multidisciplinary collaborative to collect the best and most vetted scientific research on this issue, to subject it to rigorous scientific scrutiny, organize it, and then to put it at the fingertips of healthcare workers, medical institutions, and the general public. Our goal is to provide these stakeholders with actionable information through which they can make informed decisions based on their own context, resources, and circumstances. We have no sponsor, no partisan agenda, and no other goal but to distill and present the best available and trusted science. We hope that by doing so, we can also identify the gaps in the science where further research is needed, and to fill these gaps through research carried out within our extended network and web of collaborators. So our goal today is to share this information we have about the methodologies of N95 decontamination and reuse with all of you. But most importantly, it is also to engage with all of you and listen to the open challenges that we need to move towards. While our initial focus was local, our sincere goal right now is to make our information accessible to people globally. We understand that some of the technologies we may review today may not be practical for implementation in all settings. So we want to listen carefully in order to align our future projects with problems faced in broader communities and find help solutions for these communities that capitalize on their local resources and talent. And with that, I will give the floor to my colleague, Dr. Orhun Muratoglu at the MGB COVID Innovation Center.
Thank you very much, Hannah, and welcome everyone. I'm a professor of orthopedic surgery at the Harvard Medical School, and I also run an orthopedic research laboratory at the Massachusetts General Hospital. From the onset of the pandemic, we at the Mass General Brigham have been working on extended use and reuse policies for our hospitals. The work has been carried out under the umbrella of the newly established MGB COVID Innovation Center. At the center, our mission is to organize and consolidate the rapid investigation and the clinical deployment of devices, diagnostics, data analytics, as well as therapeutics developed within our ecosystem. Next slide, please. Devices is one of the pillars of the center. And under the devices pillar, we have the reuse group that I am currently leading. This is a collaborative work with over 160 participants, some of whom actually will be presenting today. Next slide. After our initial 6 weeks of work, we have come to our own conclusions that the methods listed here in the middle, the 3 methods, are the easiest ways to implement and safely decontaminate N95 respirators. Today, you will learn about mechanism of function of N95 respirators. You will also learn about what evidence we have gathered to support the use of hydrogen peroxide vapor, germicidal ultraviolet light, and dry or humid heat. We'll tell you about the 3 main considerations to make sure that these methods can be safely used to decontaminate the respirators without compromising function. You will also learn that these respirators have a very sophisticated filter that relies on electrostatic charge, and therefore its filtration efficiency must not be compromised during decontamination. You will also learn that the fit of the respirators to the user's face is a critical consideration. Finally, you will learn that while achieving different levels of bioburden reduction, that all these methods can safely be implemented. The webinar will begin with the basics of the N95 respirators. Then we will give you the scientific evidence of why these decontamination methods are safe and effective. You will also hear from various speakers about different ways to implement these 3 methods. Our first speaker is Dr. Allison Squires, who will get us started with the basics of these respirators. Allison?
Great, thanks, Orhun. Right, so I'm an assistant professor of molecular engineering at the University of Chicago, and my job today is to make sure that we're all on the same page about N95 respirators and similar international versions, and to highlight the critical functions of these masks that actually provide protection. Next slide, please. Okay, so here are just a few examples of protective faceware that you might see people wearing, and these range widely in their complexity, the amount of protection they offer, and whether or not they can be worn more than once. On the far left, you see medical masks and cloth masks, which are basically just a barrier of material that covers the mouth and nose, but doesn't seal tightly. Medical masks are meant to be single use only, while cloth masks are washable. On the far right, you can see full face and full helmet respirators that have their own air supplies that are either purified and pumped directly to the user or are stored in a tank that's separate from ambient air. These are also generally reusable. In the center are several different models of masks that all function similarly. The ones at center right are elastomeric and meant to be reusable, and we won't talk too much about those today. But the ones in the red box at center left are N95 filtering face piece respirators and equivalent masks, which are meant to be disposable, but which are currently in short supply. This is what we mostly want to talk about today, ways to safely reuse a disposable N95 FFR in case you have no other option. Next slide, please. So we'll use the name N95 or N95 FFR, N95 mask a lot today because that's the US name for these masks, but there are many international models that are nearly equivalent and work almost in the same way. Without going into the details, I just want to emphasize that prior to selecting a respirator, users should consult their local respiratory protection regulations and requirements or check with local public health authorities for some selection guidance. And these slides will be available online in case you want to follow up on these resources later. Next slide, please. Okay, so what's in a name? In the US, N95 actually tells us exactly what this respirator does. So the N means non-oil resistant. That means that it's really good at filtering out non-oil-based particles. If you see an R or a P in the name, that means that it's either oil resistant or oil proof, respectively. The number 95 means that 95% of particles of a medium diameter of 0.3 microns are filtered out. If you saw the number 100, that means more than 99.7 filtration. And similarly, 99 is 99% filtration. Respirator implies that all inhaled air will be filtered if it's worn properly, whereas masks typically imply that there's just a barrier in place. If you see surgical, that means that there's some kind of hydrophobic splash barrier on the mask. Next slide, please. Okay, so how do N95 respirators work? Well, we know that COVID and many other viruses and pathogens can be transmitted by droplets that are present in the air around us, either that are exhaled by another person or result from coughing or sneezing. And normally you would just breathe these in and that would permit infection. But if you wear an N95 mask, next slide, please, then there are several layers protecting you from these particles. So first, an N95 respirator must seal to the face. So on the left side of the screen, you can see that yellow line indicating that the mask is really well sealed all around the nose and mouth of the wearer. There's no airflow going in or out through that seal. Second, it filters particles, and it does this with a few different layers. So first of all, we've got this outer layer, which may be fluid resistant if it's a surgical type mask, and that blocks larger particles. Then we come to a filtration layer, and that's where the majority of particles get stopped. These filtration layers we'll talk about a little bit more in just a minute. Other layers may be intended to shape and support the key functional layers. And sometimes there's a soft inner liner for comfort. Next slide, please. Okay, so a little bit more about these two key qualities, filtration and fit. So first, the way that the filter works is it's an electret filter media composed of melt-blown, non-woven materials like polypropylene. So that's a material that will trap respiratory droplets by electrostatic charge attraction. Therefore, fibers can actually have a fairly large pore size, so distance between fibers, which allows these masks to be very breathable. On the fit side, a tight seal is absolutely critical. And the next speaker will talk about this a bit, but users must every single year verify that the type of mask that they use on a regular basis fits their face correctly. Things to be aware of: if there's physical damage to the filter or a change in the filter charge, then its ability to filter out dangerous particles may change. And so this is something we really need to think about when it comes to processes that might allow the mask to be reused, but might change the filter. Similarly, if a process changes the seal, then harmful particles can leak around the edges of the mask, invalidating using the N95 in the first place. That could be caused by poor fit, facial hair, or structural degradation. Next slide, please. Okay, I want to make a really quick point about healthcare versus industrial masks. So on the left, we have a standard N95 surgical respirator that's intended for healthcare use. This is designed differently from the mask on the right that has the same filtering capability, but different hardware. So you can see this vent right on the front of it. That allows the user to directly exhale to the air. So the air that you breathe out is not filtered. So while this mask on the right might protect the user, it actually isn't great for use in healthcare settings because it doesn't protect people around you. And last slide, please. Okay, finally, I want to touch briefly on how we actually test the filtration capability of N95s in the lab. In part, this issue rises because there are so many different models of N95 and equivalent masks, and also because some of these may be counterfeit. So the standard in the US to follow is TSI 8130A, and that requires that there's a certain filtration efficiency of the filter and a certain pressure drop across that filter. The pressure drop basically tells you how easy it is to breathe. And the way that these are tested, so you can see a photo of a testing machine up here on the left, basically a piece of filter material is compressed and particles are flowed towards that filter at some standard airflow rate. Now you have a particle counter before the filter, you can see the little diagram on the right, and a particle counter after the filter. And so that makes it easy to calculate what percentage of particles have been effectively filtered out. You can also measure the pressure drop across that filter to make sure that it's quite breathable. So again, the exact standards for testing vary from country to country, and we strongly encourage you to check your local standards for guidance. All right, with that, I'd like to pass it on to our next speaker, Dr. Felicity Billings, who will talk about practical issues for how to use N95s.
Thank you, Allison. Today I'm going to be talking about the wear, reuse, and decontamination of N95s. Next slide. Next slide. Thank you. So first of all, it's important for each user to know which kind of N95 seals to their face. As Allison mentioned, the aerosol blocking ability of the N95 is predicated on the N95 actually sealing to the person's face. There are a lot of makes and models of N95s out there, so you need to figure out which one works for you. The requirement in the US is typically that annually people need to be fit tested for an N95, but this has been suspended under emergency guidance during this pandemic. However, it is still important for each user to fit test for their kind of N95 once before they start using it. The way it works is a hood is placed over your head and then aerosolized substance is sprayed into the hood. This tastes either sweet or bitter. The user is then taught how to properly put an N95 on. They put the hood back on. The aerosolized substance is sprayed into the hood again. And the fit test is passed if the user does not taste the aerosolized spray, because this shows that there's an effective seal of the mask to the user's face. Normal as well as deep breathing, some position changes are done to simulate normal wear. Next slide. Next slide. So putting on and taking off N95 is actually an important thing to go over. First of all, this is my N95. I have already labeled it on the front with my institution's coding. This is done with a Sharpie or a permanent marker prior to the first use so that I don't have to touch the front of a contaminated mask. The mask is placed into your hand and you cup it in your hand and let the straps dangle. You place the mask onto your chin and nose. Next, place the upper strap onto the top of your head and place the lower strap onto the top of your neck. You adjust the straps, and then you adjust the nose piece by pressing, not by pinching. Anytime you don an N95 mask, you have to perform a user seal check. You cover the mask with your hands and inhale sharply and make sure the mask seals to your face and you can feel that suction. You then cover it with your hands and exhale sharply and make sure that you don't feel any air rushing around the side. Once the mask is on, you then don the rest of your PPE. Doffing, which means taking the PPE off, carries a risk of self-contamination. So this is actually a very important process to get right. I put in a picture of myself in COVID intubation PPE for my hospital to show that every hospital is going to have a different set of PPE that they've purchased, and they may have different guidelines for different users doing different procedures. So make sure you know how to take off what you're wearing without contaminating yourself. The N95 is removed last outside of the patient room. Use clean gloves and don't touch the front of the N95 because it is considered contaminated. Only touch the straps behind your head. Don't pull on the sides and the front. So you take the bottom strap, pull it over the top of your head, let it dangle without touching your chest, and then take the top strap and immediately place it in a bin for decontamination or a container for reuse, and then perform hand hygiene. Next slide. Typically, N95s are considered single-use items and they're thrown away after one use. However, with current PPE shortages, many regulatory bodies, including the CDC, have given out guidance for extended use and reuse of the N95s. Extended use means wearing the same respirator for an extended period of time, and reuse means using the same respirator with multiple doffing and donning events in between. Next slide. So storage for reuse usually happens before a decontamination process is set up and can be used along with the decontamination process. You need to make sure your staff know to start saving and reusing N95s instead of throwing them in the trash, and provide them with some sort of container that they can use to keep their masks safe. Takeout containers, plastic food storage containers, or small boxes are ideal. You can see I just used a food storage container. These become contaminated, so need to be handled properly. And the important thing is that the front of the mask is contaminated and the straps are clean. So you need to be able to separate the straps on the outside of the container. Some don't save and store heavily contaminated N95s, such as after aerosol generating procedures in a known COVID positive patient. It is better to immediately decontaminate these masks before using them again. Don't share N95s between users without decontamination. Don't let the straps touch the front of the N95. And if you're using a sealed plastic container, like a food storage container, put some holes in it for airflow so you don't encourage growth of other pathogens. Next slide. So which N95s can be decontaminated once you have a process chosen? Definitely decontaminate N95s using aerosol generating procedures. I get that question a lot, and that is something that you can do. Every hospital is going to have a different level of PPE shortage, and you need to give guidance to your staff on how long to reuse N95s and how frequently to decontaminate them. Don't ever decontaminate N95s that are visibly soiled. Some methods, such as UV, can't make it through the visible soiling, and other methods have not been validated with any visible soiling. Anything with makeup, oil, such as Vaseline that people use on the bridge of their nose, blood, bodily fluids, dirt, throw those in the trash. The strap elasticity is completely necessary to have a good seal. So if the straps are too stretchy, throw those out. And any visible structural issues like tears, those should go in the trash too. Next slide. So how do you set up an infrastructure for decontamination? No matter the method, you're going to need some way to support that. Early communication is really key, and frequent communication as your process changes, because almost every hospital that's doing this has changed something along the way. Use pictures as much as you can. It's a lot easier to show people how to do things than to describe it with words like labeling masks or stacking masks. And empower your staff. People know that there's a PPE shortage and they want to help. As far as organization goes, it may seem daunting to have to set up a whole collection and delivery process, but most hospitals already have existing infrastructure for distributing clean surgical equipment or taking out the trash. Use this existing infrastructure as much as you can and retask personnel from idle departments such as canceled elective surgeries. Make sure whoever is collecting and decontaminating N95s themselves has proper PPE for the job. Use a simple mask labeling system and mark the decontamination cycle number on the N95 somewhere. Lastly, it is important to mention that you cannot combine methods. There's been no scientific evidence. There is no scientific evidence on combining methods in the same mask. So if you've decontaminated an N95 with UV, you cannot then do vaporized hydrogen peroxide on the same mask. Now I'll transition to Tyler, who's going to talk about N95 decontamination principles.
Thank you so much, Felicity, for that great explanation. I'm Tyler Chen, a PhD student and bioengineering Knight Hennessy Scholar at Stanford University. And I'm just going to walk through a little of the basics of what to consider when looking at each of these N95 decontamination protocols. So really, it's quite simple. All you need to think about is filtration, fit, bioburden, and residue. And we're going to go over these again and again in this webinar. To be an effective decontamination method, it must, first of all, preserve the N95 filtration ability for 95% of particles. Secondly, it needs to preserve the N95 fit, that tight seal to the face that Allison and Felicity emphasized earlier. Obviously, and most importantly, the decontamination method must reduce the bioburden on the mask. So kill, ideally, SARS-CoV-2 and other pathogens that may reside on that mask. And finally, it needs to not introduce additional hazards, such as hazardous residue. So starting with filtration and fit, depending on the decontamination method that is used, the number of cycles an N95 can go through before it is damaged with regards to filtration or fit will depend strongly on the N95 model and the decontamination method used. So unfortunately, even though we want to be able to say this is a one-size-fits-all protocol, it works for every mask, it works for every decontamination method, that's not the case, and it's actually quite complicated. So I would recommend you either test internally at your own facility or see n95decon.org/publications for some of the scientific studies that have been carried out on different masks for different methods. And another important point is that even without decontamination, some N95 models lose their proper fit after just putting them on 5 times. Others lose their fit after 15 times, and this has been shown in the literature. So doing that user seal check that Felicity described is very critical to ensure the mask fits after each reuse. So let's talk a little bit about that bioburden reduction. There's a hierarchy of resistance to inactivation among pathogens. And you can see near the top there are bacterial spores, which are known to be pretty tough to kill. So given that, a decontamination method that achieves 6 log reduction in viable bacterial spores can be defined as a sterilization method. Log reduction is defined as a 10-fold decrease in the number of viable bacterial spores. So 6 log reduction efficiency should ensure that out of 1 million active pathogens, it is reduced down to 1 or less. So with decontamination of N95 respirators for reuse in the emergency situation of COVID-19, our goal is primarily to, at a minimum, inactivate the SARS-CoV-2 virus, which you can see highlighted down below is a lipid virus. So it's considered to be relatively easy to decontaminate for, compared to something like bacterial spores. The FDA or the CDC recommends at least 3 log reduction against the virus for any decontamination procedure. So this is defined as being able to reduce 1,000 active viruses down to 1 or less. Processes that can achieve sterilization may be more desirable, given that what I mentioned earlier, it's very important to remember there may be other pathogens that could contaminate the mask. So certain protocols, which we'll discuss later in the webinar, may only disinfect for the SARS-CoV-2 virus, but will allow other bacteria and fungi to survive, indicating that the mask is not fully sterilized and may present other contamination risks. Next slide, please. So when validating the inactivation of SARS-CoV-2 or other viruses, there are certain methods that have been proven and tested in labs that would be ideal to execute in the hospital to ensure that sterilization is achieved. So ideally, one would validate sterilization with the appropriate bacterial spore indicators. Unfortunately, this is not always possible since some of these methods are known not to inactivate bacterial spores. So given that, it is also possible and worthwhile to demonstrate inactivation of the SARS-CoV-2 virus, which needs to be carried out in the BSL-3 facility. So this is a high biosafety level, but is ideal to determine whether or not a decontamination method is going to be effective. Alternatively, surrogate viruses can be used, such as Phi6 or MS2 phages at a lower biosafety level. And other pathogens that are found in a hospital environment should also be tested to ensure that the decontamination method is going to inactivate as many of these as possible to ensure the healthcare workers are safe. Next slide, please. So given these methods that I've talked about, there are certain methods that have been validated not to be effective, either because they damage the N95 filtration or fit, they do not inactivate the virus, or they are dangerous and leave potential residue on the mask. So of these, soap, alcohol, bleach immersion, gamma radiation have all been shown to damage the N95 filtration and should not be used for decontamination. Furthermore, storage of the mask just overnight has been shown not to sufficiently inactivate the virus. That is not enough time to inactivate the virus on the surface. Secondly, UVA or B solutions, such as nail salon products, they are also not sufficient, do not deliver a sufficient UVC dose in order to inactivate the virus. And similarly, sunlight has not been proven to inactivate the virus due to too low of a UVC dose. One important point is that bringing potentially biohazardous masks home is highly dangerous and carries significant contamination risk. So decontamination should only occur in a secure setting, such as a hospital or a third-party provider. And finally, yeah, thank you for listening. This is basically the summary of what to think about when you're looking at any of these methods the next presenters are going to discuss. Filtration efficiency and fit are very important and highly method dependent, depending on the decontamination method that is used. Bioburden inactivation must demonstrate at least a 3-log reduction of SARS-CoV-2 on the surface of the N95, and preferably sterilization methods should be used that achieve 6-log reduction of bacterial spores as well to ensure that other pathogens are not going to be contaminating the mask. And finally, hazardous residues on the surface of the mask should be minimized. Thank you very much, and I'm going to pass it on to our moderators for a quick Q&A.
Great.
Thank you so much to all of the panelists for their talks. Great. Thank you so much for all the panelists for all their talks. A reminder to attendees that you can vote on questions that are in the Q&A. So there's a little button on the kind of bottom of the screen that will say Q&A, and we would love for you to vote on questions there. Please also take a moment to answer the poll questions that are popping up on your screen if you can. So now we're going to take a few questions. The first question that we have here is for Allison. Allison, is there a quick and dirty way to determine if N95s are safe post-decontamination?
That's a really great question. Unfortunately, no, there's not a quick and dirty way to know whether or not a decontamination method is completely safe. And that's exactly why N95DECON and MGB have been working so hard to go through all of the available data. The 3 critical elements for safe reuse are 1, inactivation of the virus and ideally other pathogens. 2, the mask must retain its ability to filter particles so that the user is still protected. And 3, the mask must retain its fit so that it still seals to the user's face. The only one of these things that can be tested on the macroscopic level with some quick and dirty check is really the seal to the face. But the other 2 things, knowing that the virus is inactivated and knowing that you haven't altered the material properties on the nanoscale of that filter that protects you, those are things that have to be tested in the lab. So first of all, you need studies for specific mask models to make sure that a particular process is safe, inactivates virus, and doesn't change the ability of that mask model to work. And second, you can actually check the filtration capabilities of different masks after a process has been done, just to verify that it's working correctly and hasn't changed the ability of the mask to filter. Thanks.
Thank you so much, Allison. Mark, do we have time for one more question?
Great. Yeah. Looking at the poll, we're already seeing that there's a wide range of time and frequency at which various workers are able to receive N95 masks. And so there's great pressure on how you can extend the life, for instance, of an N95 mask as well as decontaminating it. And one of the questions we've often gotten asked on this is, can you use something like a surgical mask over your N95 mask to protect it? And make that a more effective way to wind up utilizing it. I'm wondering if Felicity might be able to comment on that a little bit.
Yeah, this is a frequent practice that people do. Unfortunately, surgical mask supply is also limited. So I wouldn't encourage people to use surgical masks plus an N95 in most circumstances. When you're donning and doffing your PPE, like I mentioned, there's a high risk of self-contamination every time you're taking something off. So if you do use a second mask, make sure that you're removing that properly also. The CDC recommends in its extended use recommendations to use a cleanable face shield instead of a second mask over an N95.
Terrific. Thank you very much. So we've received, again, the poll. Thank you for introducing this information and getting some feedback to us. Broadly, we're getting questions on what the most effective way to decontaminate the N95 masks are. So with that, we're going to move on to the next section of our discussion here today, and kick off with Jill Crittenden, to share with us some of the methods of decontamination. Jill?
Hello, and first I would like to thank the healthcare workers who are the real heroes against this pandemic. I'm a research scientist at MIT, and the project that I worked on for this was to do a critical review of hydrogen peroxide as a method for decontamination of the N95s and to communicate the data that are known to healthcare workers. The next slide, please. This slide shows a detailed list of the numerous methods that are available for hydrogen peroxide decontamination of the N95s as well as other PPE. And I'd like to just spend a couple minutes on it because it highlights important differences. So in general, the method is to use concentrated hydrogen peroxide or hydrogen peroxide with additives and to convert that into a vapor or an aerosol with a machine, and then that vapor or aerosol covers all portions of the N95 mask and decontaminates them. And the important aspect of that is that it needs to be aerated completely before it's breathed through, because the hydrogen peroxide mist is dangerous to breathe and also creates irritants on the skin and the eyes. And so a fully contained chamber is an important aspect of this, as well as the full aeration. And depending on the method used, the amount of time required for complete aeration of the mask also varies. And so I want to emphasize that different protocols need to be used for the different methods. One difference that I would also like to highlight is that one of the methods applies an electric field to generate plasma, ionized plasma from the vaporized hydrogen peroxide. This is an advantage in that it aerates it much more quickly, but that method can damage the filtration properties of the mask. And so if those machines are used, the STERRAD machines, proper protocol needs to be used to not repeat the method too many times or use the standard setting on your machine. It needs to be an express cycle or a cycle specifically described by the company. Another important difference is liquid hydrogen peroxide, which is a method that was investigated some years ago where the N95 mask is submerged in 6% hydrogen peroxide for 30 minutes. And that is effective in terms of not disrupting the filtration. It's currently under investigation for complete decontamination. And also the amount of time required to aerate after such a submersion also has to be tested. Next slide, please. So advantages of the hydrogen peroxide method are that it's an extreme sterilant. So in a bottle, hydrogen peroxide is not reactive, but when it interacts with biological materials, it creates free oxygen radicals that destroy these materials, including lipid membranes. So the SARS-CoV-2 virus is enveloped by a lipid membrane that's destroyed by the hydrogen peroxide. It also destroys DNA and RNA and proteins. And so it's very effective at decontamination. It's also good because it doesn't require heat to decontaminate things. And so it can be used on plastics or anything that's heat sensitive. Furthermore, it can penetrate a lot of spaces because the vapor just settles inside and outside of any surfaces that are small or dark. And finally, the breakdown products are not harmful, unlike ethylene oxide gas, for example. This just breaks down into water and oxygen. The next slide, please. So disadvantages that I already brought up is that hydrogen peroxide is a severe respiratory hazard. And so it needs to be, these machines and methods need to be done with care by trained personnel. It often requires equipment that's expensive. Many of those machines cost many thousands of dollars and need to be used by trained personnel. Some of them can be rented. And the equipment specific protocols, so you can't just use the same protocol for different machines. And another point is that this method is not compatible with cellulose. And so you need to ensure that the mask you're using does not contain cellulose. And on the N95DECON.org site, we've tried to make a comprehensive list of all the N95s that do and don't contain cellulose. The next slide, please. So as I mentioned, it's very effective at killing pathogens and viruses, including some of the most hardy pathogens, for example, anthrax spores and C. diff spores. C. diff spores, for example, that might also be in your hospital environment and that you want to be sure that in case your mask was exposed, it's also decontaminated of those spores. And the details references for this decontamination can be found on the N95DECON site. Next slide. Also contains a number of references that you can find the details for on the N95DECON site. Fit and filtration are not heavily impacted by hydrogen peroxide. So the one outstanding example of that, as I mentioned, is the STERAD system, where if the improper protocol is used, then filtration can be disrupted. And so you need to pay special attention with that one. But the other systems are not known to disrupt fit or filtration. Next slide, please. So in summary, the bioburden reduction with this method is excellent. However, to make sure that adequate hydrogen peroxide was used, a biological indicator or at least a chemical indicator should be used for each cycle. The N95 performance is also excellent with most of the methods. Check the cycle number and the protocol to be used, and also be sure that your mask does not contain cellulose. Other concerns are that 2 of the methods have additives. It's not known whether those additives, and you can see that by checking back on the first slide, are respiratory hazards, but also it has not been tested. And so there's no expectation of problem, but that is a potential concern. Some of the methods are consistent with having the N95s in a Tyvek pouch, methods that use the plasma, and others are not. So that's another consideration. And also the amount of time for aeration before it can be returned to the healthcare worker to breathe through safely is important to check for each. To implement these systems in your hospital, you need to be sure that you have safety trained personnel, the appropriate equipment. And if you want to try to set up a new system, for example, in a whole room, please check the hospital protocols that we've provided links to these on our N95DECON site. And also the US government has provided a checklist to determine whether a new room in your hospital is compatible with hydrogen peroxide, depending on how much, for example, the size of the room and whether it contains materials that can absorb the hydrogen peroxide. So thank you very much for that. And with this, I'll pass it on to Dr. Orhun Muratoglu, who is going to talk about implementation of the hydrogen peroxide methods in the hospital setting.
Thank you very much, Jill. And many thanks for the scientific evidence that you provided to us that supports the use of hydrogen peroxide vapor to decontaminate the N95 masks. I'm now going to tell you more about the implementation of this method. Next slide. So far, this method has been implemented in many institutions around the United States. Duke Medical Center is using the BioQuell system. Both the University of Iowa and the Brigham and Women's Hospital in Boston are using the SteraMist. And finally, at Mass General Hospital, my home institution, we implemented the use of STERIS hydrogen peroxide vapor generator. Next slide. So for this technique, we are using a room that has its own dedicated ventilation system. The hydrogen peroxide generator that you see in the picture is placed in the room. The respirators are placed on shelves or hung freely. Please do not stretch the elastic bands on N95s, as this would adversely affect the fit on the user's face later on. So we make sure that the gas can access both the inside and the outside of the respirators, so we do not block the respirators in any way. And we fill the room with the hydrogen peroxide gas from the generator in about 15 minutes to reach a level of approximately 500 parts per million of hydrogen peroxide. And we maintain this level for 3 hours, which is then followed by degassing or aeration until the count of hydrogen peroxide in room air is under 1 part per million. Next slide. We use chemical indicators and biological indicators in different locations of the room. Chemical indicators allow us to determine if the gas is uniformly distributed in the room, and the biological indicators allow us to determine the extent of bioburden kill. At 500 parts per million of gas for 3 hours, we have uniform gas distribution and 6 logs of reduction in bacterial indicators during this decontamination process. Next slide. As Jill showed you earlier, the hydrogen peroxide vapor does not compromise the filtration efficiency even up to 20 cycles of decontamination. And the same is true for our system that we're using in the hospital. Next slide. Next slide. Yes, the respirators that we have processed in our room have all passed the fit test after decontamination. And finally, there was no significant residual hydrogen peroxide on any of the masks after decontamination. Next slide. In addition to the bacterial spore testing to ensure that the bioburden reduction is reduced, we also investigated SARS-CoV-2 and showed 4 logs of virus reduction on masks that were contaminated with the virus. Next slide, please. So in conclusion, a vapor hydrogen peroxide method can be implemented either with the use of a room or maybe in a small biosafety cabinet. About 500 parts per million of the gas at 3 hours of exposure is sufficient to safely and effectively decontaminate the N95 respirators. Next slide. In Boston, we are now using a larger scale hydrogen peroxide vapor decontamination provided by Battelle Memorial Institute. The concept is the same as the one I just outlined and can decontaminate 80,000 masks per day. We use fine point Sharpie markers to mark the masks, decontaminate them, and then return them back to their original users after each decontamination cycle. We also have an ongoing post-decontamination surveillance program. So far, up to 2 cycles of use followed by decontamination, we did not see any adverse effects on mask function in terms of filtration and fit. Now let's switch gears and hear from Dr. Samantha Grist about evidence that supports the use of germicidal UVC. Samantha?
Thank you very much. My name is Samantha Grist. I'm from the Bioengineering Department at UC Berkeley, and today I'll be presenting on the evidence for UVC decontamination of N95s on behalf of the UVC team of N95DECON. First, I'd really just like to thank all of the healthcare workers, first responders, and everyone else on the front lines of this crisis. Now, when we speak of UV or ultraviolet decontamination, we're specifically referring to the germicidal UVC region of the electromagnetic spectrum, which is shown on this slide. Looking at the spectrum, the different colors or wavelengths of visible light are shown on the right. And as we move down to lower wavelengths or higher energies of light, we pass through the non-germicidal UVA region, the minimally germicidal UVB region, and then the germicidal UVC wavelength range. At wavelengths lower than that, the light can actually generate ozone from air, which is a health hazard. Now, you might wonder why the UVC region is germicidal. And this is shown in the germicidal action spectrum below the EM spectrum. This plot shows how effective light is at inactivating pathogens as a function of its wavelength. And you can see there's a peak in the spectrum at around 260 nanometers. This is because UVC inactivates pathogens by damaging their genomic material, and DNA and RNA have maximum absorption at around 260 nanometers. This is why this germicidal UVC range is typically used for decontamination. Next slide, please. Within this germicidal UVC range, the evidence indicates that an irradiation dose of at least a minimum of 1 joule per centimeter squared inactivates viruses similar to SARS-CoV-2 on N95 materials. Dose represents the integrated intensity of light that's delivered to the N95 and is the crucial factor for decontamination. It's important to note 3 things about these data. First, while this dose yielded a 3-log reduction in active virus on the majority of tested N95 models, it was not sufficient for all of them. 3-log reduction means that for every 1,000 starting active virus particles, 999 will be inactivated. Secondly, the N95 straps were not found to be as effectively decontaminated with UVC as the face pieces. And so the straps require a secondary decontamination method, such as wiping with a compatible disinfectant. The third important point is that this minimum dose of greater than 1 joule per centimeter squared is much higher than doses that are needed for surface decontamination, so surface protocols are not going to be sufficient. Pathogens on N95 material can need 1,000 times the dose that they need on surfaces when they're inactivated on N95 materials. And this is thought to be due to the attenuation of UVC as it passes through the layers of the N95. So higher doses are required to bring some decontamination to those inner layers. Next slide, please. Now, at these high UVC doses, it's logical to wonder whether we're damaging the N95 materials. And there's also evidence from the literature that suggests that N95s keep their fit and filter performance after 10 to 20 cycles of UVC decontamination. N95 damage was observed at higher doses, equivalent to about 100 decontamination cycles. And so with that in mind, we anticipate that the limiting factor for reuse is actually going to be fit degradation from donning and doffing alone. And that's because fit degradation to below necessary standards is going to be highly dependent on the N95 model, and it's been observed after 5 to 15 donning and doffing cycles with no decontamination at all. All the evidence summarized here, as well as additional sources, are available in our N95DECON technical report on UVC. Next slide, please. I wanted to highlight here some important implementation concerns. First, there are several types of sources that are not going to be appropriate for UVC decontamination. There are sources that, while they might output the correct germicidal UVC range of wavelengths, they don't have sufficient power or uniformity, or they don't have enough bulbs or reflectance to mitigate shadowing issues within the treatment area. Shadowing is a huge concern because UVC decontamination can only inactivate pathogens that see that minimal dose of germicidal light. At lower wavelengths, as I mentioned, the light can generate ozone from air. And this ozone, unlike the risks from the light itself, does not go away when the light is turned off. Other sources like tanning beds, nail polish curing lamps, and sunlight by the time it reaches the earth are not anticipated to generate sufficient germicidal radiation for N95 decontamination. As the decontamination efficacy is directly tied to the dose and thus the source, as well as the N95 configuration with respect to that source, a crucial step in setting up any UVC treatment protocol is to validate that the minimally acceptable dose of 1 joule per centimeter squared reaches all of the surfaces of all of the treated N95s within the treatment range. And this needs to be performed with a calibrated sensor specific to that UVC germicidal wavelength range. It's also important to note that environmental control might be important. There's evidence that higher humidity results in less effective decontamination with UVC. And finally, implementation of UVC, like any decontamination approach for N95s requires a robust industrial hygiene workflow for implementation. And as UVC does not sterilize the N95s, each N95 has to be returned to its original user. It's only decontamination, not sterilization. Next slide, please. So to summarize, the evidence suggests that a germicidal UVC dose of at least a minimum of 1 joule per centimeter squared delivered to all surfaces is likely to sufficiently inactivate viruses similar to SARS-CoV-2 on N95s, although this bioburden reduction depends on the N95 model. This dose is absolutely crucial to decontamination, so it needs to be validated with a calibrated UVC specific sensor. It's important to note again that the straps require secondary decontamination and that this dose is not likely to inactivate all other pathogens throughout the layers of the N95 because different pathogens can have different robustness to UVC. So each N95 should be returned to its original user and not cross-contaminated with other N95s during this process. At these doses, from the evidence, we expect that the limiting factor for N95 longevity is going to be fit degradation from donning and doffing alone. It's also important to note that UVC is hazardous to human cells, just as it is harmful to pathogens. So appropriate engineering controls and PPE are crucial to not expose any humans or people to UVC. Additionally, several UV sources, including home UV, sunlight, and sources outputting low wavelength UVC are not appropriate for UVC decontamination. Next slide, please. And now to speak about turning this evidence into implementation, I'd like to introduce Dr. Thomas Baer, who will present the first part of this section on UVC implementation. Thanks.
Thank you, Samantha, for that excellent overview of UVC decontamination methods. UVC exposure has been used effectively in many decontamination applications, but instruments designed specifically for decontamination of N95 masks or other personal protection equipment are not generally available. This has led several groups to begin developing custom UVC equipment specifically for this purpose. We will now discuss design principles that should be considered, some basic safety precautions, and 2 examples of custom UVC decontamination chambers. N95 mask decontamination requires illumination of the entire mask surface. Next slide, please. At the accepted level for decontamination of 1 joule per centimeter squared. This requires that the optical design of any device illuminate the mask surface as uniformly as possible. Since the mask has 2 sides and several layers, it is important to expose both front and back surfaces of the mask to the UVC light. The N95 mask is a 3-dimensional object with significant surface curvature, so it is optimal to illuminate the mask from all directions. The most common UVC source is a germicidal lamp. It is important to select germicidal lamps that are not ozone producing. This is particularly important if the design incorporates an enclosed chamber, since the amount of ozone that builds up within this enclosed space could be significant. Non-ozone producing UVC lamps do not appear to generate a significant amount of ozone, but if you can smell ozone, it is best to avoid breathing the air in the chamber when it's first opened immediately after the decontamination cycle. UVC light damages human tissue and can damage skin and eyes, so direct exposure should be avoided. Next slide, please. There are 2 types of UVC sources that are generally used for decontamination procedures. UV light emitting diodes, which usually emit at around 270 nanometers, and mercury discharge germicidal lamps that primarily emit at 254 nanometers. Germicidal lamps have been used for many years for decontamination, and there are industry standards for different lamp geometries and output powers. LEDs have been available for only a few years, and to my knowledge, at this point in time, industry standard device geometries have not been developed. There are a number of trade-offs between these 2 sources, which are listed on this slide. In general, germicidal lamps are much less expensive for any given power level and have acceptable lifetimes of several thousand hours. 2 examples we will present in this webinar both incorporate commercially available germicidal lamps. Next slide, please. The first design, developed by a group at Stanford University, incorporates 16 30-watt UVC germicidal mercury discharge lamps inside of a standard metal storage cabinet with dimensions of approximately 180 centimeters tall by 90 centimeters wide by 60 centimeters deep. That's 6 feet by 3 feet by 2 feet for our US listeners. The inside of the cabinet is lined with standard household heavy-duty aluminum foil, which has a reflectivity of over 80% at UVC wavelengths. The foil lining increases the overall intensity incident on the masks and also provides illumination from all angles. The lamps are about 90 centimeters long and are arranged in banks on the cabinet doors and backside of the cabinet. They are fastened to the cabinet in linear arrays with a lamp spacing of about 23.5 centimeters. Our optical models predicted very uniform isotropic illumination of the masks, which is consistent with our measurements. 30 to 50 masks can be loaded onto a frame and exposed in a single batch. The time required to reach the exposure level of 1 joule per square centimeter is under 5 minutes. One of the goals for this design was to use components that should be available in resource-limited settings and could be locally sourced. This design can provide sufficient throughput to satisfy the daily needs of most hospitals. The design is open source and details will be made available through the N95DECON website. As a side note, next slide, please. I built this unit in my garage at my house using just a battery-powered drill and hand tools, and it took me only about 2 days. Next, Dr. Martin Persky from Massachusetts General Hospital will discuss their UVC decontamination chamber design and how they are planning to implement this approach into the hospital workflow. Martin?
Thank you very much, Tom and Samantha, for the kind introduction and for the great overview about the UVC panel. So our second unit was developed by MGH and Harvard University and is a little bit different. It has a horizontal operating design and it's a smaller unit. It was designed to be a point of care, fast decontamination unit for, in particular, small hospitals and remote facilities which can't use the mass decontamination units. Next slide, please. So it's enclosed. It's about 3 feet by 2 feet in length and width and depth. It's covered with aluminum foil to increase reflectivity and to get a uniform irradiance. It has 12 standard low pressure mercury germicidal lamps, 6 on the top panel and 6 in the bottom panel. It has near uniformity. It has some safety features which we incorporated. There's no UVC leakage. There's a door interlock so that there's no way that UVC can exit the chamber once operating, once you open the door. And there's an automatic timer shut off to make sure that the masks are not getting overexposed. And it's very user-friendly. It's a one-button operating system, so to turn it off and turn it off, on and off automatically. Next slide, please. So we did the performance testing and we showed on the left side the irradiance and showed that it's a uniform uniform irradiance throughout the box. We did the mass fit testing and the performance test as well. And on top, we did 2 bacterial spore testings and showed on the bottom right that we could see that our unit kills a 6 log, up to 6 log bacteria spore after 1 joule per centimeter square. Next slide, please. So I think Samantha and Tom mentioned the shadowing, and we try to address it by placing UVC intensity labels on our mask in different positions. You can see on the left top side, the outside and the inside of the mask, and the yellow dots are, or stickers are, the UVC sensitive labels. So on the right, you see the outcome. So in a control unexposed, you see the yellow color. And once we expose the labels with UVC, it turns more and more green to a saturated green and a 1 joule per square centimeter irradiation on the bottom line. All of our sites were fully exposed, except this one site 7, which is underneath the strap, where the strap is stapled to the mask, where you see a little of a yellow shadowing. So, but that in mind, we can't change this one. It's due to the design of the mask. We see barely shadowing except on the location 7. Then the other question was as well if our unit itself creates some shadowing. So we have like this mesh tray where we put our masks on, as you can see on the bottom left picture. And this metal could also induce some shadowing. So we did a low exposure of 0.1 joule per square centimeter square where you could see some shadowing on a far left picture, like with this hexagon lines in yellow. But once we reach our 1 joule per square centimeter squared, we see a complete irradiation as well. And we assume there's a maximum of a 5 to 10% reduction where these grids are. Next slide, please. So I want to talk about implementation a little bit, how we assume it will happen in our hospital. First of all, before bringing the mask to the decontamination unit, you should write your name or any kind of identifier on your mask with a permanent sharper marker, and inspect the respirator for any kind of decontamination or surgical spills or bigger like damages, and then take them out. For the procedure, we should place 1 mask in a bag or in a plastic container and then send it to the unit. Next slides, please. So for the decontamination unit, you always should wear eye and skin protection, instead like for example, goggles or gloves. For the procedure, you open your unit, you pull out the tray, avoid any shadowing by putting the mask on it one by one. You shouldn't overlap them. Then remove your gloves, close the unit, and start the decontamination cycle for 2 minutes. After the 2 minutes, the unit shuts off automatically. You re-glove, open the door, pull out the tray, and then remove the mask, mark them as decontaminated, and place them in a new user-labeled paper bag or container for pickup. Next slide, please. What we saw so far, there are a lot of institutions using UVC already. Some use like contained boxes like Tom and I produced, but some use like these whole room decontamination processes, which are meant for decontaminating rooms in general, but not necessarily for UVC. And these procedures have some issues if they're not used in a proper way. As you can see on the right side, they are masks stacked behind each other. So, and as Samantha mentioned, they have to be really see the UVC light, otherwise they won't get the proper irradiation dose. And some masks, as you see on the right picture, like the industrial mask, have like these plastic filter on top, which we mentioned before earlier in a webinar. And these masks wouldn't penetrate necessarily at the filter material, the mask. So next slide, please. I want to hand it over to the heat group. And thank you very much.
Thank you. Hello, my name is John Doyle, professor of physics at Harvard University. It's a pleasure for me to discuss heat decontamination of N95. And I would like to thank the previous speakers. They laid out the key issues of viral inactivation, fit infiltration, and other key issues such as return to index user procedures. Next slide, please. It's well known that high temperatures inactivate viruses and bacteria. At high enough temperatures, all viruses and bacteria will be inactivated on all surfaces, including inside, for example, a N95 filter material. However, at too high a temperature, the N95 may fail due to changes in the N95 materials. At low enough temperature, the N95 will still function very well, but the virus may not be inactivated. Based on current science and literature, it looks very likely that there is a range of temperature where the virus and many co-pathogens will be inactivated while still leaving the N95 functioning well. There is also some evidence that moisture added during the heating process can aid in viral inactivation. Next slide, please. The current evidence specifically for CoV-2 virus indicates that under lab conditions of a temperature of 70 C, 60 minutes, with no added moisture, this is also called dry heat, when you don't add any additional moisture. So with no added moisture in these dry heat conditions, this will inactivate the CoV-2 virus to a level a bit better than the FDA guidelines of 3 log reduction. We also note that the higher temperatures used in autoclave process of 121 C, already known to be a sterilization process in general, meaning that it inactivates bacterial spores, as was discussed earlier. that this temperature also strongly inactivates CoV-2 virus. We emphasize here that the extensive work on inactivation of viruses in liquids and protein-laden media should not be used as a specific guide for heat inactivation of CoV-2 on N95. In particular, one should not use the 60C 30-minute guideline for decontamination of N95. Furthermore, 70C 60-minute heat treatment has not yet been shown to be sufficient in a clinical setting. Finally, temperatures between 70 and 85 C will not inactivate all possible bacteria and spores, and because of this, return to index user procedure should be used. Next slide, please. Regarding the functionality of the N95, both fit and filtration must be considered. Current evidence is listed in the bottom half of this slide, but I direct your immediate attention to the top half. In summary, many N95 will still function well, passing both fit and filtration tests when processed at a temperature of 85 C with 80% relative humidity for 30 minutes. In contrast, some N95 will not function if brought to a much higher temperature of 121 C, autoclave temperatures. The next speaker will expand on these points more extensively. However, some N95, in particular some pleated masks, will function after autoclave treatment. It is heavily model dependent. In summary, evidence is building that a temperature between 70 and 85 C under certain conditions will work well for decontamination. But currently, data on CoV-2 inactivation is still limited. These temperatures will not inactivate all other possible pathogens. As will be described by the next speaker, the essential approach is very simple in concept. N95 is indexed to user by using labeling the N95 with name or other identifying information. The N95 is placed in a container and the container is placed in an oven. However, in order to ensure that the right temperature and possibly humidity, it may be that humidity is important for the process, to ensure that the right temperature and humidity level is reached, independent sensing should be employed. I would like to strongly emphasize that inactivation of CoV-2 virus in this temperature range is a very active area of research and new results are on their way. Thank you. I'd like to pass this on to the next speaker. Cole Eisenhelder.
All right, thank you, John. I am a physics graduate student at Harvard University. I have been working with N95DECON on heat decontamination methods. I'm going to talk about specific considerations for implementation of 3 heat methods, and then the specific method that we have developed in our group. Next slide, please. The first method I'll talk about is autoclaving, which is attractive for 2 reasons. One is that it is widely available in many settings. And the second, as John mentioned, this is already known to be a sterilization technique. Unfortunately, as John noted, there is an extreme model dependence that has been observed, where molded N95 models, such as the 3M1860, have been shown to fail quantitative fit tests after as little as 1 to 2 cycles. However, pleated models such as the 3M1870 have been shown to pass quantitative fit tests for at least 5 treatments. So if considering autoclaving, it is very important to understand whether the specific mass models in question have been validated and are compatible with this process. Additionally, it should be noted that up to now, few studies have actually included filtration efficiency. And so this is an ongoing area of research where we need more information. Next slide. The next method is microwave generated steam, which has been recommended by the CDC as an option for decontamination. Unfortunately, while there is data showing inactivation with other surrogate viruses, at this time there is no data indicating that there is an activation of coronaviruses. Additionally, the number of test cycles that have been used in studies up to now have been very limited. Mask models have been shown to survive 1 cycle, with some mask models surviving 3, but few studies have gone beyond that. Additionally, there is a question of whether or not filtration will survive after 5 cycles in steam. Finally, there is an open concern with metal components posing a sparking hazard in microwaves. Current data indicate that this is unlikely to be a concern, however, should be considered carefully in any methodology that is considered. Next slide. The last method I'll talk about is humid or dry heat conditions. And as John noted, there is a careful window that needs to be considered where if you go to too low of a temperature, you run the risk of not inactivating the virus. And if you go to too high of a temperature, you run the risk of damaging the fit or filtration of the mask. And so in any method using heat in this way is very important to make sure that you are carefully monitoring and validating that you are achieving the desired temperature and humidity for the desired time. Additionally, this is not a sterilization method, and so it is very important to both prevent cross-contamination of masks and to use an index user return method so that no masks are shared between users. The last point to make is that the N95 mask is made up of multiple materials, and at the low end of the recommended temperature range of 70 C, there is evidence that there may be less inactivation on the metal nose piece. So there is specific considerations that should be looked at there for secondary decontamination. Next slide, please. Now I'll talk about our specific method that we've developed, which uses Ziploc containers to contain masks, along with a wetted paper towel for humidity control. And then these masks are then transferred to a forced air convection oven for heating, and then they can be returned to the user. Next slide. In this method, we're specifically targeting 85 degrees C and 60 to 80% relative humidity. It's important to note that we've used forced air convection ovens here, unlike many ovens. There is no direct line of sight to heating elements with these ovens, so there is no risk of overheating the masks and developing hot spots in your oven. The containers we've chosen to use here are very beneficial for two reasons. The first is that they prevent cross-contamination without potentially crushing the masks. And they also provide a fixed rigid volume that can be used for humidity control. And we do this by calibrating the volume of water that is placed on the paper towel to this volume of the container. And with this, we've shown that we can easily achieve the desired temperature and humidity for the time we're looking at. And after this process, there's a necessary 5-minute drying step to make sure that the mask returns to ambient humidity. This method is very scalable, as with a 3.7 cubic foot oven that we used in our testing, you could very readily perform decontamination of more than 1,000 N95s per day. Next slide. As noted, though, it's very important to validate and monitor your system to make sure that you are achieving the target temperature and humidity for the desired amount of time. We do this using electronic sensors that are capable of operating in the desired range. It's important to consider whether or not your sensors are capable of this, as not all sensors can achieve both temperature and humidity simultaneously. It's also important not to rely on an oven thermostat, as it may be offset from the actual temperature seen by the masks and may not represent whether or not there are cold spots or hot spots in your oven. To check for this, you should validate this in multiple locations, as we've shown in this image, with sensors in multiple boxes. Additionally, you want to determine the warm-up time, as in our system, there's about a 10-minute time to warm up before you're actually at your target temperature and humidity. And so you need to consider that in your total cycle time. Next slide. And now we have Andrew Barnard talking about another heat implementation method.
Hi, I'm Andrew Barnard. I'm an associate professor of mechanical engineering and director of the Great Lakes Research Center at Michigan Tech University. I'm going to talk to you today about our heat implementation on a larger scale. Next slide. So our goal was to work with an industrial partner and build a PPE decontamination unit to help during the COVID-19 pandemic, primarily considering the facts listed here. We wanted to make something that was mobile, that could be used in many locations, that was made from readily available components that could be easily purchased all over the world, was relatively cost effective, easy and safe to use, and especially scalable. We designed a very large scale system, but everything we did here was designed to be scalable to smaller scale systems to right size this for your facility. Next slide. So what we did is we took a 40-foot insulated container. This is a refrigerated shipping container that was previously used to ship vegetables overseas. We took the refrigeration unit out and turned it into a heated chamber. We did that using residential components. So we used 3 residential heating units, residential blowers, heaters, and ductwork in order to circulate the air. And air circulation is key in this implementation to make sure you get the same temperature throughout the entire volume of the container. For control, we use residential thermostats. And to move PPE in and out of the system, we use industrial baking racks, things that are readily available and relatively inexpensive. In fact, we just drove around our town, collected baking racks from businesses that weren't using them to do our tests. Next slide. You can see that we're able to heat up the entire chamber relatively quickly. It takes about a 40-minute preheat cycle. Then we can hold that as long as necessary. This data shows holding that temperature for about 50 minutes after we've run our preheat. On the right, you can see some thermal images just showing that the temperatures before and after the cycle are vastly different. Next slide. So having made a dry heat chamber and having more literature come out saying that moist heat may indeed provide better performance, our second generation was made by our industrial partner, Thermotronics, and incorporated both programmable heat and humidity control. Here you can see an N95 mask in a test container with a data logger inside of it that logs humidity and temperature. And on the plot, you can see the temperatures and humidities that we're reaching. And in this test case, we were trying to control at 80% humidity for 30 minutes. And you can see at that high of a humidity, we can control very accurately, but it does take 2 to 3 hours to get the chamber up to those conditions before you can start your soak cycle. Next slide. It's very important to think about procedures when you're doing this. Our system is currently undergoing emergency use authorization paperwork through the FDA. And the FDA is very concerned about getting everyone's masks back to the original user, which we've spoken about before in this webinar, but also avoiding cross-contamination between masks on racks and between racks as you move them throughout a hospital setting. So what you see here is very small text. Please don't try to read the text. This is our preliminary document that shows how you might use our system from donning and doffing the mask to bagging it, to putting it on a rack, then bagging those racks as you walk them through the hospital, removing those bags when they're in the container, cleaning them or disposing of the bags, running the system, and then changing labeling, rebagging the racks, and returning to the users who put the masks on the shelves. So these types of procedures should really be thought through if you're going to use the heat and humidity method, because it is only a virucidal decontamination method. Next slide. So in conclusions for the heat implementation, you need to carefully control the heat and humidity, and you need to measure this throughout the chamber to make sure it's evenly distributed. This is achievable with ovens and is very easily scalable. We've shown 2 scales here, both a benchtop scale system as well as a large system that can decon up to 5,000 masks in a couple hours. But this is not a sterilization technique. This is strictly virucidal decontamination. We would caution against autoclave and microwave steam at this point. From autoclave, the concern is that the molded N95s will not pass fit check afterwards. And with microwave steam, there's just not enough data out there yet to be able to say anything about that. So with that, I would like to turn it over to Orhan for some concluding remarks.
Thank you very much. And well, I hope that we were able to convey to you that these 3 methods listed in the middle of the slide are the safe and effective methods to decontaminate N95 masks without compromising filtration efficiency or the fit of the respirators, and that the extent of bioburden reduction achieved with these methods is acceptable. The slideshows in this webinar will be made public shortly, and the YouTube live feed will be available for later viewing. So now I would like to thank all of our collaborators at N95DECON and MGB ReUse workgroup for their invaluable contributions to our understanding of N95 masks, and the speakers for their contributions to this webinar. And I would like to now open this webinar to questions and answers. Mark?
Great. Thank you very much, Orhun. And welcome back, everybody. We really appreciate your participation with us on this. We've got a number of questions that have come in the Q&A, and please continue to contribute those. And then going through them, one of the areas that people have asked is essentially that we've got lovely technologies that we're discussing here, but in areas where there's low resource settings, for instance, how does one actually decontaminate masks in those environments? And in particular, how long would you need to just store a mask, for instance, for a virus to be inactivated before it gets worn again? So can we ask for example, Tyler, perhaps to answer that question for us? Can one just store a mask for a period of time and then safely reuse it?
Yeah, sure. Happy to. Thank you, Mark. If you could go to the next slide, please. We've got a slide prepared on this one, actually. Sorry, one more. Yeah, perfect. So if there's no other choice, the CDC has listed storing an N95 at room temperature for multiple days as a method for possible decontamination for SARS-CoV-2. Unfortunately, there's very little data on how long an N95 needs to be left at room temperature to kill the SARS-CoV-2 virus. And it seems that the survival of the virus is highly dependent on the surface on which it is inoculated. From the few data points we do have, though, it seems that storing it in a clean, breathable container at room temperature for 7 days may inactivate the virus, whereas storing just overnight is certainly not sufficient to inactivate. This method should only be used, again, if there is no other choice for decontamination, since just the time method will not protect the wearer against bacteria or mold that may be on the N95. And finally, it's very important that the N95 is used by the original owner to maintain fit and prevent cross-contamination, given that other pathogens may survive this method.
Great. Thank you very much, Tyler. Really appreciate it. I'm going to point out to all of our participants today, we have a poll going on right now. This will be up for a little bit, really to get a sense of what the areas are that you're looking at. And it's nice to see that there's a broad balance of interest out there as well. And this points out that there is a need for all of these techniques. So with that, Marianne, can you take the next question?
Yeah, I have a question for Hannah, please. Hannah, is this group planning to set up a collaborative platform where we can share projects and knowledge?
This is a fantastic question and suggestion. I think we should consider that very, very seriously, especially as we move more into researching and implementation in low and moderate resource settings. So thank you, whoever asked this question, thank you for that and for the suggestion. I would like to take actually the opportunity to answer another question that was in the Q&A, which is about whether we're gonna post these slides online. And the answer is yes, please visit our website for these slides if you're interested in that.
All right, I have a question here for John, please. John, can PPE be decontaminated with dry heat at, for instance, 125 F for 1 week? Yeah.
Thank you. That's an excellent question. I would first re-emphasize what Tyler said about time method by putting the mask in a container for 7 days. This is also covered at the N95DECON website about the key considerations. And so person asking the question quite rightly thinks, well, okay, if we hold a mask for 7 days at room temperature, we get a certain amount of inactivation that, on the mask, that if we raise that temperature to say 125 F, which is about 50 C, that you should get even better inactivation. And the science would indicate that that's true. Unfortunately, there is no research that I'm aware of that has been done to actually study inactivation of the virus in that temperature and time window. So it's for future studies, and at this point, no specific recommendation can be made.
Thank you so much. Back to you, Mark.
Great, thank you. Our next question will go to Jill. We're getting a number of questions on the types of materials that can be used for masks. And in particular, this discussion of this electrostatics in an N95 or a P95 mask. Can you explain a little bit about what that is, and then more broadly about how you can think about that relative to different materials for used in especially low resource settings?
Yeah, the electrostatic aspect of the N95 filtration turns out to be particularly important because it allows for there to be good airflow, because the particles, respiratory particles, are trapped by an electrostatic charge rather than simply a small bore that the respiratory droplet can't make its way through. And it's been shown many years ago, maybe in the 1980s, that disruption of that electrostatic capacity of the masks with ethanol, for example, it can also be disrupted by submersion in soap, and also exposure to gamma rays or other ionizing rays. All of those things can strip the N95 of its charge. And as previously shown, that has a major impact on the filtration ability of the mask. So extra care needs to be taken to not disrupt that electrostatic property of the mask. And hydrogen peroxide wetting, for example, those things do not strip it of its charge. The other thing that's known to strip that charge is severe agitation and high heat. So those are not recommended protocols.
Great, thank you. Marianne.
Okay, I have a question for Samantha. Samantha, there have been lots of questions about validation when a UVC sensor is not available. Can you use a UVC dose strip dosimeter? Would that be adequate to measure the energy generated?
Thanks. Yes, validation is absolutely crucial. Regarding the photochromic sensors, there's two, I guess, potential issues with these sensors. The first is that the commercial UVC photochromic strip sensors that we've tested are designed for surface decontamination, and they can saturate after doses of 100 to 400 millijoules per centimeter squared, which is not sufficient to show that minimum of 1 joule per centimeter squared. The second issue is that these color strips might overestimate UVC dose, because even the UVC strips can actually be sensitive to UVB as well as UVC, and UVB has much lower germicidal efficacy than UVC. And so because of these issues, we suggest currently, wherever possible, to use a calibrated UVC specific sensor to measure dose. But Dr. Martin Puszka does show these strips in his talk as a secondary validation method. And we're also conducting further characterization of these strips to suggest potential protocols. If these strips are used, extreme care must be taken to validate that the appropriate dose is reached. It would be necessary to multiply the treatment time by the appropriate factor to reach that 1 joule per centimeter squared critical value. Plus, we would also suggest including a large safety factor in that calculation, because that bulb light output can actually vary significantly during exposure. Thanks.
Thank you so much, Samantha. Back to you, Mark.
Great. Tyler, we're getting a number of questions from people about other types of masks, whether they're KN95s or other that are out there. There are a lot of masks out there. What do you need to consider in the decontamination of those? Now, the methods that are shown here applicable in those areas?
Absolutely. Thank you, Mark. That's a great question. So unfortunately, as you mentioned earlier, it seems that these decontamination methods, especially the heat method, they have very different effects based on the N95 model that is used. So we expect transitioning from an N95 to a different mask entirely, something like a surgical mask or a KN95, which have been shown to have different construction, may not necessarily have the same properties. So if there's a method that's been validated for a certain N95 model, it is, I think, safe to assume that if that method is carried out properly, it will continue to perform well on that model. But for other models, there's a lot more that needs to be tested. So you can't necessarily just go straight into a new model and assume that, hey, this is going to be all fine, because there are differences in construction. On our website at N95DECON.org, in the appendices of our technical reports, we have a list of a large number of N95 models and which ones have been tested for which methods. KN95 data we do not have yet. So as data comes out, we'll be adding those to our website.
Great, thank you very much. Marianne?
Yeah. Hi, I have a question for Cole. When using the heat decontamination method, does the plastic straps on the N95 mask get damaged?
Yeah, thanks for the question. That's a good question to ask about any of the different materials in the masks. For the elastic specifically, there is some evidence that if the elastic is stretched while the masks are still hot, that it can be expanded and damaged. But we've seen that if you allow the masks to cool before donning them or stretching the elastic, then there is no evidence that we've seen of any damage to the elastic at the level where it would impact the fit of the mask, which is what really matters.
Back to you. Yeah.
Thank you very much. So next question is going to be for Orhun. One of the questions people have asked is, first of all, where's the availability of things like the Battelle process out there? And a number of people have asked that question. And with that, for places that may not have access to the Battelle process, that's a hydrogen peroxide vapor overall, how can they utilize the existing systems they might have to safely accomplish the same decontamination?
Thank you, Mark. Battelle is unfortunately only available in the United States, and there will be approximately, I think they're allowed to build up to 60 units. So they will dispatch 60 units around the country, and each one of these units will be able to decontaminate up to 80,000 masks per day. Outside the United States, Battelle is not available, and therefore we recommend that you use one of the hydrogen peroxide generators that might be available to you. If you have a generator, then the rest is going to be pretty straightforward. You either can use a biosafety cabinet, basically a small cabinet that you can use to hang the N95 masks, and fill this cabinet with the hydrogen peroxide vapor for a certain duration, maybe the way that I described earlier at 500 parts per million for 3 hours, followed by several hours of aeration to make sure that the gas concentration goes down to 1 ppm. And this can be detected with handheld detectors very easily. Another approach, as I described earlier, is to use a room that you can seal and maybe close the ventilators. If the ventilation of the room is not specific or is not exclusive to that room, then you can maybe block the ventilation in the room and use that room to do the same process. Either way, you will have very good results in decontaminating the masks and reducing the bioburden, not only with the virus, but also with the other pathogens that might be present on the masks. Now, this method is very good to return the masks back to anonymous users, but currently in Boston, we are returning them back to the index users.
Great, thank you very much, Orhun. Marianne.
Yes, I have a question here for Tom. This question is from an attendee who has purchased a number of CPAP UVC cleaners for family members who work as medical professionals. This person has purchased a product by Lumens that claims to clean CPAP masks with UVC at the 254 nanometer range. Do you have any comment on this?
Thank you, Marianne. I'm not familiar with that specific decontamination product. However, there are several cautions. There are considerable difference between a CPAP mask and the N95, as Samantha has described. There are many layers in an N95 mask. And so the amount of fluents necessary, the joules per square centimeter, would be significantly higher in for an N95 mask as opposed to a CPAP mask. As I mentioned, you need to illuminate from both sides. So you'd have to make sure that the mask was illuminated, the instrument illuminated from both sides, and it'd be best if it's an isotropic illumination where there's illumination from all angles as well. So I would be extremely cautious about using this, and there would be no way to really know how long the exposure levels would need to be for an N95. So in general, I would say it's probably not a good idea and probably should not be used.
Great, thank you so much, Tom. Mark?
Great, I'm gonna go right back to Tom on this in a second. For those of you who may know, this webinar is also being broadcast on YouTube Live, and we have a question from one of our YouTube Live viewers as well, saying that they're interested in using UV LEDs as a source for decontamination, and if there's any sort of studies that Tom knows of looking at ultraviolet LEDs that might be applicable to this area.
Yeah, thank you, Mark. The UV LEDs are potentially as effective or even slightly more effective than germicidal lamps, since they typically emit more at the peak of the decontamination wavelength, 270 nanometers, as opposed to the germicidal lamps that emit at 254. They're both quite effective and can be used, and there are a number of groups that have looked at using these in the LED sources as decontamination. Part of the complications are there are no industry standards for manufacturing specific device geometries for the UV LED, so they will all have to be characterized based on the specific design. The germicidal lamps tend to have more of a standard format and output, and tend to have a industry standard that they all conform to. So they're a little bit more predictable in terms of the performance levels that you would expect. There's also issues associated with lifetime, which are unknown with LEDs, power cycling, in both LEDs and germicidal lamps for that matter. In general, the germicidal lamps are better characterized for this application at this point in time. Ultimately, the UV LEDs are going to be a very, very viable alternative, but there is still new enough in the commercial marketplace to be a little bit unpredictable as how well they're going to perform under these circumstances.
Great, thank you. Marianne?
Thanks. I have a question for Tyler here. Tyler, there have been some questions about people wanting to decontaminate N95 masks using ozone. Do you have any comment on the use of ozone for decontamination? And also, is this method potentially harmful for patients due to the ozone exposure due to poor health effects from ozone exposure?
Yeah, fantastic question. So ozone has not yet been fully validated for N95 fit, filtration, or the inactivation of SARS-CoV-2. So at this point, the ozone method is entirely unproven, given the literature that we've seen so far. We do have a team of scientists monitoring that literature, though. So as new studies come out with regards to ozone's effects on N95 fit, filtration, and the inactivation of SARS-CoV-2, we will be updating them on our website. Given the question of ozone exposure, yes, that's correct. Ozone is dangerous, although it depends on the concentration. So after a certain amount of time of degassing and reaction, ozone may be reduced to low enough levels that it is safe. But during an actual ozone treatment, certainly that is something that would be a health hazard to consider.
Thank you so much, Tyler. Mark?
Great. I'm going to come back to Tyler again on this next question, because we have another question from YouTube Live. Because there's a number of techniques, especially in low resource settings, where you're able to decontaminate other medical equipment, things like boiling in water for 10 minutes. I wonder if you can comment on that, or perhaps leaving things out in sunlight, what those efficacies are.
Yeah, thanks, Mark. So yes, this is true. I know a lot of the methods that we've talked about here are tailored for settings that have the funding to buy these large scale equipment, and that's something that we're working as hard as we can to find methods that are also going to be valuable for lower resource settings. Given that there is not much data out there to support whether or not some of these methods will work. For example, boiling or putting a mask in boiling water has not yet been tested for, again, filtration, fit, or inactivation of SARS-CoV-2. For lower resource settings, some promising methods include the heat method that's been described, the time method, which requires no resources except a clean place to put the mask. And finally, there are some early results that are showing liquid hydrogen peroxide, 6% liquid hydrogen peroxide immersion as being a potential method of inactivating SARS-CoV-2 on the surface. Although, again, this has not yet been fully tested. And there are some concerns about bubbles that can form around the surface of the hydrophobic mask that may prevent the virus from being inactivated. So these are things that are all being explored, and we will update them on our website as soon as we have data. And this is really a focus for us. So if folks have more comments or questions on this, please feel free to send us an email or reach out to us, because we're happy to help.
Great, thank you. Marianne?
Yeah, thank you. I have a question now for Andrew. Andrew, isolation gowns are currently in critically short supply worldwide. What do you think about decontaminating isolation gowns as well in the type of setup that you have made?
Yeah, that's a great question, and one we've been thinking about too. I mean, we're seeing isolation gown shortages here in Michigan right now as well. So one of the reasons that we scaled our device the way we did, being very large, is so that down the road we could do more than just masks. We're focusing on masks right now because that is the immediate need, but we see needs arising for things like gowns or even larger scale hospital equipment like gurneys. But all of that would need to be tested. There's been no testing done on any of that. So I would say until the test results are in, don't do that, but it is certainly an active line of research that we're interested in looking into.
Great.
Thank you so much, Andrew. Mark, next question.
Yeah. Thank you. Hey, I'm going to ask a question back to Samantha. Looking at some of the techniques that are used, for instance, can they be used for decontaminating small lots of masks? For instance, the same way they do in a dental office or dental practice.
Thanks. So I think there's a few really important points to highlight here. First, not all ultraviolet is the same. As I mentioned, you want the bulb to emit in that germicidal UVC range rather than UVA or UVB. So you should verify that the equipment does that. And additionally, that it's not in the ozone generating range. So always check the manufacturer's specifications for this information. Shadowing and irradiance are also a concern. Sometimes these kinds of equipment might only have one bulb and they might not have sufficient reflective materials. And so there can be non-uniformity, significant non-uniformity and shadows within the device, so that not all sort of sides of that 3D N95 topography might be effectively treated. Not all surfaces are given that effective dose. And so to know whether the bulb is working for decontamination, you really need to validate that UVC dose. And the other important point that I would like to mention for any of these smaller scale systems is that even in a smaller scale system, this really, any UVC implementation or any N95 decontamination implementation really has to be accompanied with a robust industrial hygiene workflow to make sure that those N95s are processed appropriately and not cross-contaminated. And so that workflow needs to be set up. Additionally, those types of devices might only irradiate from one side, and that can actually introduce additional concerns as well. The N95s will have to be flipped, and you need to make sure that you don't cross-contaminate the mask with that flipping process. For example, by flipping onto the same surface that the dirty side of the N95 was previously on. So we do recommend wherever possible to irradiate from both sides at the same time and to be very careful with that industrial hygiene workflow. Thanks.
Great. Thank you very much. Marianne.
Thank you. I have a question now for Orhun. Orhun, does pressurized hydrogen peroxide vapor or ozone speed up the decontamination process?
I just answered that question online also. I guess just briefly, it possibly could. We have not looked into this. There are machines out there where you can actually pull vacuum inside the chamber where the masks are sitting and then backfill the chamber with the hydrogen peroxide vapor. That would probably increase the penetration of the gas into the nooks and crannies of the mask. So therefore maybe a little faster than the regular methods that we're using. However, these chambers are quite expensive and we don't have access to one. We used one of them actually at one of our industrial partners. And we decided that that will be a very expensive method for us, considering that we need to decontaminate 25,000 masks every week.
Thank you very much. Mark?
Great. I'm going to go back to sort of relative to low resource settings, particularly in the heat method for Andrew out there. You know, it's wonderful you've shown the system that you've stood up, but if I don't have a capability to even building that level of a system, how do I wind up getting appropriate instrumentation? How would I set it up so I actually establish an ability to safely decontaminate my masks in my area? I wonder if either you or John could answer that. Start with Andrew.
Sure, I'll take a shot and then turn it over to John. There's lots of ways to create heat. We chose electric heat because in our setting here in Michigan, that's ineffective way to make heat. But we also looked at things like steam generators and other types of heat sources that may be more efficient in other parts of the world. Certainly, resources are going to be an issue, and things like finding a 40-foot refrigerated container in some parts of the world are going to be very difficult to do. Really, all you need is a box that you can insulate, and you just need to insulate it so that you can prevent that heat from leaking to the outside. So any type of insulated box is sufficient for the exterior. Any type of heating is sufficient for the interior that allows you to circulate that air throughout the entire chamber so that it can be evenly heated. But there does need to be some sort of measurement effort to make sure that that heat is getting evenly distributed throughout the chamber. And that could even be as easy as placing thermometers within the chamber and checking them immediately upon completion of the cycle. So it doesn't have to be a very sophisticated thermocouple based system. It could be using traditional temperature measurement techniques. So John, you don't want to add anything to that?
I think that that was very much summed it up. The key points being that if you have a device, an oven, a box that's got additional heaters, however you were able to provide the right temperature without direct line of sight to the heating element, and with flow of air within the device to evenly heat the N95 in containers, then it's an effective way of getting those N95 up to the right temperature for decontamination. And so there's many different ways of doing this. There's many devices. One can even imagine using ovens that exist in hospitals already that are have the right temperature range, and there's work being done to try to implement some of those at different locations within the United States. So as long as you've got the right temperature, follow these other guidelines, then that N95 will come up to the temperature that you're targeting.
Great, thank you very much. Marianne.
Yeah, I now have a question for Tyler about writing on the surface of N95s. We've had some people ask about whether writing on the surface of an N95 damages the material since the solvent can be alcohol.
Yeah, that's such a good question, and I'm really happy that people are asking that, and I'm really sorry to say that we don't actually know. I've seen no studies yet on the effect of writing with a permanent marker on the surface of the N95. I know that there are many hospitals in the US today that are doing that, writing on the surface, but there are also other hospitals that have pointed that out as a concern and have instead elected to label the straps of the N95. So yeah, unfortunately, we don't know. That data, I think, is not yet been tested. But if anyone out there is running these tests or has that data, please send it to us, and we will make sure to highlight that on our website because it's a very important question, given that so many folks are considering how to label.
Great.
Back to you, Mark.
Okay, thanks. I'm going to ask a question back to Orhun because we've gotten a number of variations of these questions relative to hospital settings. First of all, how often are the people in your hospital essentially decontaminating their mask or using their mask? What duration? And are there any sort of procedures that can be applied to, say, rotating masks less frequently, using surgical masks, for instance, with non-critical patients, or are there other, operational ways to wind up decreasing the frequency at which decontamination is necessary?
Well, there are some reuse and extended use policies that one can follow. In our hospital right now, we're using N95 masks exclusively whenever we're taking care of COVID patients in the ICU and other settings. And those masks are decontaminated using the Battelle system, which is the hydrogen peroxide vapor system. We decided that even though Battelle is safe up to 20 cycles of decontamination, we decided that we will only go up to 5 cycles of decontamination because with our stock of N95 masks at the hospital, 5 cycles will be sufficient for us to be able to maintain enough supply of the N95s to continue to survive during this surge. And And as I said in my talk earlier, we are continuing to assess the changes, potential changes to the masks during the decontamination process, because all the data that's been published, as you have to realize, has been with brand new masks that are decontaminated and tested for filtration and fit. And that has been done several times with no actual use in between. In our post-surveillance program, where we are collecting masks that are actually used in the clinical setting, decontaminating them so we can see the true effects of use plus decontamination. And after 2 cycles, we have not seen any change in the filtration efficiency or fit test. And we're continuing our surveillance program. We will go up to 5 cycles and then we will stop at that point.
Great, thank you. Marianne?
Great. I right now have a question for Martin. So Samantha described some of the smaller UV boxes, but can you address more broadly the logistics of decontaminating masks in clinics, fire stations, police stations, and other small offices?
Yeah. So right now, I think there's no smaller unit or like a validated unit available as per se, but we heard there's a couple of companies right now looking into it to build like smaller, more like really top and bottom irradiation units, which are smaller in size than ours or Tom's, and which can be then put into an office in a fire station, and then you can decontaminate 2, 3, 4, 5 masks at once, which is sufficient for smaller institutions or offices or fire departments. But it's in the process and they will be evaluating it. And yeah, they're coming up probably in the next couple of weeks with it.
Thanks so much, Martin.
Hey, I'm going to ask a question now for Samantha. Looking at the ways of decontaminating with light, you hear about things like sunlight and visible. One question we've had before is, what if you use infrared, since heat matters? What if you use infrared light on these materials? And do you have any data or studies on this?
Unfortunately, I'm not aware of any evidence, any peer-reviewed studies looking at infrared specifically, or combinations of infrared and ultraviolet or UVC for decontamination. This could potentially be an area for further research, and I guess if that is performed, I guess we understand that both the bioburden reduction and then also the N95 performance need to be evaluated in response to that new treatment or those combination treatments. Thanks.
Great, thank you very much. Marianne?
Yeah, Cole, I actually wonder if you have some additional comments on that from the heat perspective. Do you have any additional comments on infrared heating?
Yeah, I think infrared heating, if used from a heat perspective, should be considered with caution, as it will be unlikely to provide the same uniformity of heating that you can get with something like a good forced air convection oven. And as was mentioned earlier by Andrew and John, it's really important that for any heat method, you're making sure that you are really achieving the correct heat level with your system. So that could be considered, but it would need careful study.
Thank you so much, Cole. Mark, do you want to ask one more question?
Great. Well, first of all, I want to thank people that have joined us here today. The poll is still up. It's very clear that a lot of the heat methods are important that people wind up looking at. We're trying to wind up providing enough information to get back to you on this. I guess one of the last questions, I'm going to bring this back to Orhun on this, saying with hydrogen peroxide, the liquid sprayed hydrogen peroxide onto a mask, does that wind up working?
That's a good question, Mark, and we have not looked into this. My first reaction is going to be, we're not going to recommend using hydrogen peroxide liquid because it will leave behind residue on the mask. And given the fact that the healthcare workers are breathing through the mask, we don't want to have them breathing these hydrogen peroxide particles that are left as residue on the mask. So I would say no.
Great, thank you, Orhun. So I think we're coming towards the end of our time here. First of all, I want to thank all of the participants, especially the frontline healthcare workers that may be on this webinar that are joining us today. Very critical. Your time is really important. We appreciate it today and hope this information is of use to you. We'll be sharing all the information from this in terms of a link to the YouTube Live, as well as the answers and the slides that are provided. So with that, I think we can wind up turning this over to finish up. So thank you very much.
Fantastic. Thank